It's all yours.
Thanks a lot.
From TNO.
Yeah.
TNO is a new partner in the consortium. Very happy to have you on board, and I'm very looking forward to the research that you're going to do the next few years.
Thanks a lot for the introduction. So my name is Arnout Mieremet, and I'm really happy to be here. So thanks all online as well to be here, and also live in Amsterdam. So today I will tell about the project that was funded by ZonMw, about testing of personalized interventions, targeting restoration of the gut microbiome in ME/CFS. So a little bit of background information, because this is the first time I can present this project in this meeting. So the gut microbiome is part of the dysbiosis in the gut microbiome is part of the whole of the pathogenesis in ME/CFS, which is complex, and several mechanisms play an important role, as also something we are experiencing today with all the different topics covered in the different presentations. But the gut microbiome dysbiosis is considered as a key factor contributing to the disease development. And key microbiome readouts are affected in ME/CFS, and this is something that was shown in several research projects that were published in literature. For example, the diversity in the microbiome is reduced, which means that the composition in the microbiome is changed, and also the metabolites that are produced by the microbiome in the gut are affected in ME/CFS. So this also leads to a potential target for treatment. And for example, gut-targeted treatments might be able to relieve parts of the symptoms in ME/CFS. However, clinical studies are expensive and can be a burden for the patients. So therefore, using in vitro models, so lab models, are attractive alternatives to test for first treatment effects. And for example, in this project, we focus on development of in vitro gut microbiome models, but also other models representing other parts of the symptoms are very interesting. So the aim of the current project is to develop and apply functional in vitro models of the ME/CFS gut microbiome. And with these non-animal models, interventions aiming to improve the gut microbiome and its interaction with the intestinal tissue can be tested. So we are making use mainly of two types of models. So the first type of model is the iScreen model, so the in vitro screen, which is a high throughput assay that mimics the colonic microbiota. So from patients participating in NMCB, fecal material is collected, and it's collected in such a way that the microbiome can be preserved after freezing once or twice, and then it can be cultured in our lab for 24 hours or 48 hours, depending on the assay. And in that assay, in the iScreen, these interventions can be tested. And a recent publication of our lab showed that when we observe an intervention effect in vitro, this correlates significantly with observations in also in vivo in clinical studies. So that means our platform is really predictive for in vivo clinical effects. Another type of model is called the intestine in vitro model. And with this model, ex vivo intestinal tissue can be obtained and cultured for a limited time. And with this in vitro model, the interaction between the metabolites that are produced by the gut microbiome and maybe inflammatory effects on the intestinal epithelial tissue can be tested in such a high throughput assay. And with this assay, we aim to understand what's the effect on barrier formation or barrier functionality in the gut. So in this project, we divided all tasks in different work packages. So in the first work package that we're currently executing, we aim to include patients in this shared cohort between ME/CFS lines and NMCB. But also we aim to include ME/CFS patients and healthy controls in the cohort of only NMCB. And we aim to collect tubes for fecal collections and also tubes for setting up our in vitro models. And we aim to test, that's the main aim of work package 1, the collection of the materials. Then the main aim of work package 2 is to develop these in vitro models and aim to understand better what we can observe in vitro. And then the main aim of work package 3 is to combine observations of our in vitro models with in vivo observations and make use hopefully of complex data analysis to really show associations between what we observe in vivo and what is observed in vitro. So in this project, we collaborate with representatives of 3 patient organizations, and we also collaborate then, as mentioned, with MECFS Lines and NMCB and other scientific collaborators. And within TNO, we also collaborate with different departments. It's a real team effort here as well. So thank you all for your attention, and if there are questions, please let me know.
What are these, or what were these in vitro models used for before?
Yeah, so before we could, for example, model the effects of different interventions, let's say fiber interventions in healthy population to understand what's the effect of specific fibers or fiber combinations in general in the gut microbiome and also in this intestine model system. Other questions?
You're saying significant differences were found, but how large? Are they likely to be clinically relevant?
I'm not exactly sure which significant results were mentioned of the interventions. So the study that was recently published in December of 2025 showed a significant correlation between the observations we make in our in vitro model with in vivo clinical outcomes. That means when we have an exposure in our in vitro system for 24 hours, that predicts an effect also of in vivo within the gut microbiome that is affecting after 8 or 12 weeks. So it's really a predictive model for in vitro that predicts in vivo outcomes.
Yeah, last one. How do you determine what interventions to test? Do you look at patient experiences too?
Yeah, so that is something we are open for more input based on the input of patient representatives from the different organizations to really pinpoint which intervention we can introduce in our in vitro test system. Although we have a preference for a combination of testing prebiotics and probiotics and a combination thereof.